Hello everyone, welcome to my channel Biochemistry Insights. So we have already discussed about light harvesting complexes in the photosystem I and photosystem II, how they participate in photosynthesis in our part 1 photosynthesis video. This is the second part of photosynthesis video, and here we are going to discuss about the light reactions of photosynthesis. There will be a third part of photosynthesis video lecture wherein we will be covering the dark reactions. So let us move on to the overview of photosynthesis. So photosynthesis is the process in which green plants and algae convert light energy from the sun into chemical energy and store it as sugars. So this is what we know about photosynthesis. Now the entire thing, the conversion, trapping of sunlight and conversion into sugar is actually done in two different phases. First is light phase that occurs during daytime in the grana of the chloroplast. Then comes a dark phase reactions that occur during day and night both, but the location is stroma of the chloroplast. In this video, we will be concentrating on the light phase reactions, which will involve photolysis of water and photophosphorylation. So as I said, photosynthesis, light phase reactions are also known as Hill's reaction. This involves photolysis of water, photophosphorylation that leads to formation of NADPH and ATP. Dark phase is also called Kelvin cycle, and this leads to fixation of carbon dioxide and then assimilation into sugar. So chloroplasts in the mesophyll cells of the plant leaf are the site of photosynthesis, and these are the stacks of thylakoid known as grana, and in these granal stacks, thylakoid membrane, the photosynthesis occurs, especially the light reactions. The dark reactions occur in the fluid part of the thylakoid portion or the chloroplast, which is known as stroma. We have already discussed how light harvesting pigments are arranged in photosystem II, that the reaction center is P680. Photosystem I, the reaction center is pigment 700. Both these photosystems, they differ in the arrangement and types of pigments found in them. Carotenoids, chlorophyll B, chlorophyll A are part of photosystem II with reaction center P680. Chlorophyll B, chlorophyll A are part of photosystem I with reaction center P700. So these pigments, they trap sun's energy, and they activate the reaction center chlorophyll. The excited reaction center chlorophyll loses its electron to the primary acceptor. Okay. Now let us understand how these photosystems work. So this is a typical picture of a representative picture of thylakoid membrane, and supposing this is photosystem II. So we have reaction center here down towards the thylakoid luminal side. We have the reaction center, and the primary electron acceptor of photosystem will be on the stromal side of the membrane. Okay, and in between the electrons will be circulated from the light harvesting pigment to the reaction center. Reaction center will get activated. The excited electrons will be donated to the primary electron acceptor. And towards the luminal side of the thylakoid membrane, what will happen? Photolysis of water will happen, releasing electrons that will fulfill the loss of electron from the reaction center. So this is the normal process. Now let us dig into the details. Let us now understand the entire process. In this picture, you can see two sides. One is thylakoid lumen, that means the inner side of the thylakoid. And this is the upper portion represents the chloroplast stroma, the fluid portion outside the granule stack. And clearly we have this is your thylakoid membrane. Now thylakoid membrane, inside the membrane we have photosystem II and the electron carriers which participate in the transfer of electron from photosystem II to photosystem I. Then we have some electron carriers which take up electrons from photosystem I and finally they reduce NADP and form NADPH. So we have electron transfer from PS2 through various electron acceptors, then it falls in the PS1 and then again few more electron transporters are involved and finally NADPH is produced. Okay, now you must be wondering what is this big thing here. This is enzyme ATP synthase. As the name suggests, this is the enzyme which phosphorylates ADP into ATP. It has two components. One is the membrane component, which acts as a channel protein, which allows protons to move through it from one side of the thylakoid membrane to the other side. And this is the flower-shaped subunits. These are the catalytic center which phosphorylates ADP into ATP. Now let us understand what happens here. So, when photosystem II light harvesting complexes, they become excited. The photons are transferred to the reaction center and the reaction center chlorophyll becomes excited, loses its electron to the primary acceptor pheophytin. Pheophytin transfers electron to the next member in the chain, that is plastoquinone. While plastoquinone accepts electron from pheophytin, it also takes up two protons from the stroma. This is very important here to remember this, that plastoquinone also takes a proton from the stroma and becomes PQH2. That means it got reduced. And now it transfers electron to the next member, that is cytochrome, but protons it releases into the luminal side. So what has happened here is protons from the chloroplast stroma were taken up by plastoquinone and protons, where does the proton land up? Proton gets accumulated in the luminal side of the thylakoid membrane. अब इलेक्ट्रॉन के साथ में क्या हुआ? What has happened to the electron? Electrons got transferred to cytochrome. Now cytochrome transfers electron to the next member, that is plastocyanine. Now plastocyanine is that component which can transfer electrons directly to the reaction center of photosystem I. Now the photosystem I reaction center chlorophyll becomes excited, loses its electron to the primary acceptor, the ferredoxin. Reduced ferredoxin transfers the electron through the various iron sulfur center present in the thing, and then finally it transfers the electron to the ferredoxin NADP reductase or FNR complex. This FNR complex reduces NADP and forms NADPH on the stromal side of the chloroplast. Okay. So this is the first product of your light reaction, that is phosphorylation, photophosphorylation. The first product is NADPH, which has formed by the non-cyclic transfer of electrons from photosystem II to photosystem I and then finally to ferredoxin NADP reductase that reduces NADP to NADPH. Now, while doing this electron transfer, I told you to remember that we are actually transferring protons from stromal part and accumulating it in the luminal side. So, why are we building this proton concentration gradient? So, after some time, what will happen? The concentration of protons is inside thylakoid lumen is more, and this enzyme ATP synthase opens up its membrane component, and because of the proton motive force, it opens up and protons will move forcefully through the enzyme complex and generating a proton motive force. Because of the proton motive force, the energy of proton motive force is utilized to phosphorylate ADP into ATP. Here, the phosphate is obtained from the stromal milieu. That means inorganic phosphate from the stroma is utilized to phosphorylate ADP into ATP. And energy we are obtaining from the proton motive force that generated because of the electron transfer between PS2 and PS1. Therefore, now we have two products. One is NADPH, second one is ATP. NADPH got formed because of the electron transfer and ATP formed because of the proton motive force generated because of the electron transfer. Okay, so this is how transfer of electrons is also responsible for the formation of ATP. So let us recall one more time. Excited electrons from the reaction center plus protons from the stroma are transferred to plastoquinone. Plastoquinone becomes PQH2. That means it is now reduced. Cytochromes accept electrons from plastoquinone. Protons are released into the thylakoid lumen. Therefore, along with the electron transfer, what is happening is protons are getting accumulated in the thylakoid lumen, creating a proton gradient. Eventually, concentration of the protons inside the thylakoid lumen becomes very high compared to stroma. So, in a way, you have created a gradient of protons, just like a dam on a river which holds water on one side, the other side is deficient in water. Whenever we require, we open the door and the water rushes through that gate. Similarly, here also across the thylakoid membrane, protons are stored on one side only. The other side we have a lower concentration of proton. Okay. This is another way of showing a simplified form of non-cyclic electron transfer between photosystem II through pheophytin, plastoquinone, cytochrome, plastocyanine, P700, ferredoxin, then the FNR complex and finally leading to production of NADPH. Herein, there is one thing which I want you to understand. There is one mineral or element that is very important when it comes to photosynthesis, and that is manganese. What is the role of manganese in photosynthesis? Manganese is very critically involved in photosynthesis, specifically in photosystem II during water splitting or photolysis of the reaction. Now you must be wondering, where does the photolysis happen? Photolysis happen at the reaction center of the photosystem II. Water is split into protons and electrons and oxygen is given out as a byproduct. Why this photolysis happen? Because photolysis is that reaction which funnels back electron lost by the photosystem II reaction center. So if you stop photolysis of water, the lysis of water is stopped, then you cannot regenerate the electrons lost by the photosystem II reaction center. So this photolysis of water is very important as far as photosynthesis is concerned. Now for this photolysis to happen, manganese is required. So now you understand how crucial is manganese for photosynthesis. Now, location in the photosynthetic apparatus. Where does this photolysis happen and where is your manganese present? Manganese is found inside oxygen evolving complex or OEC or water oxidizing complex, which is part of PS2. OEC is located in the luminal side of the thylakoid membrane in chloroplast. Structurally, catalytic core is Mn4Ca O5 cluster, a metal cluster containing 4 manganese ions, 1 calcium and 5 oxygen atoms. So, at the water oxidizing complex of PS2, we have Mn4Ca O5 cluster. Okay. I hope this is clear. So, this is the situation. This is supposing the Mn4Ca O5 complex, where it is at the reaction center of near to the reaction center of the pigment safety photosystem II in the thylakoid membrane. Right? The manganese enables oxidation of water molecules into oxygen, proton and electron. The energy of proton excites photosystem's reaction center chlorophyll. This energy is used to remove electrons from water in a stepwise oxidation process at the manganese cluster that we saw in the previous slide. Manganese ions cycle through multiple oxidation states to accumulate enough redox potential to split water. The overall reaction is shown here. 2 molecules of water is split. The electrons liberated is held by the manganese cluster and oxygen is evolved. Let us now understand the mechanism how manganese functions in photolysis of water. This is called Koch cycle, also called as S-state cycle, and it describes oxygen evolving complexes stepwise oxidation process. So, OEC undergoes oxidation from S0 to S4 states, where each step represents one photochemical charge separation and manganese oxidation event. At S4, the cluster has accumulated enough oxidative power to extract 4 electrons from 2 water molecules. So, water loses 4 electrons and releasing oxygen. Calcium in the cluster provides structural stability, but manganese is the actual redox active center. Now, why manganese is essential? Only manganese can provide necessary multi-electron redox chemistry under biological conditions. Without manganese, photosystem II cannot oxidize water, meaning oxygen will not be produced. No proton gradient will be built and no electron supply to the photosynthetic electron transport chain either. Therefore, photolysis of water is central to the entire light reaction, and that photolysis of water is dependent on the manganese calcium complex, and that manganese multiple oxidation state is responsible for the photolysis of water and evolution of oxygen. So, we have now completed the light phase reaction. Just to summarize, light phase reactions are also known as Hill's reaction. It has two parts, photolysis of water and photophosphorylation. During photolysis of water, two molecules of water is split. 4 electrons are extracted from water, which are held by manganese calcium complex, and those electrons are funneled back to the reaction center of photosystem II. Photosystem II reaction center accepts photons from the trapped sunlight, becomes excited, loses its electron to the primary electron acceptor, transfer of electron from primary electron acceptors to photosystem II, and then finally reduction of NADP to NADPH forms the proton motive force across the thylakoid membrane, and movement of proton motive force and associated photophosphorylation results in the generation of ATP. So we have two products from light reaction, NADPH and ATP. In our next video, we'll understand how the products of light reactions are funneled into the dark phase reaction, which is also known as Calvin cycle, and details of the Calvin cycle. Thank you. The next upcoming video will be photosynthesis part 3, which will cover the dark phase reactions. Keep watching.